Today we're talking about mutations. So we first need to define mutation. A mutation is any change in a DNA. It doesn't need to be harmful. It doesn't necessarily need to cause a changed amino acid. It just needs to be a change in the DNA. So in this example here, we have TTC, and it codes for the mRNA of AAG, and it codes for lysine. Here, if it's changed to TTT, then it goes to AAA. It still codes for lysine. So because it codes for the same amino acid, we say that this is a silent mutation. So some mutations can cause no change. Some mutations, however, do cause a change. So a small change, if it swaps out one letter, would be called a substitution. So instead of AAA, it might have AUA, and then it might change the amino acid. And this is called a substitution. We're substituting one letter for another. You can see here's a substitution. Our original sequence is TGG, CAG. Here we just switched out one letter, the C for the T. Okay, so usually we're only changing one amino acid here. Sometimes we can get a large change, either a deletion or an insertion. An insertion is when we insert some letters, we add some extra ones. And a lot of times what happens is it shifts all these letters over. And so they might not code for the same codons anymore after that. Same thing with a deletion. Here we see that two of the letters were deleted. Again, it's going to shift over all of those letters so they no longer code for the same amino acids after that deletion. So what can cause some mutations? We can have mutations caused by mistakes in DNA copying. So interphase, when we've got helicase and DNA polymerase making copies. Radiation, so like X-rays or sunlight. Chemicals, right, in cigarette smoke or cleaning products. And then also some viruses and bacteria can cause mutations.